Organoids are a pioneering technology in the medical, veterinary, and life sciences. The easiest way to think of an organoid is as a miniature version of an organ tissue that can be grown in the lab. The backbone of organoid biotechnology is the stem cell. Stem cells are a progenitor cell type that can be stimulated to differentiate into diverse cell types. In the correct growth conditions, stem cells can be made to differentiate into specific cell types that make up a particular tissue. Scientists can use this approach to facilitate the stem cell-derived growth of three-dimensional, multicellular, self-organizing cellular structures termed organoids. At the Moredun Research Institute, we have been developing species and tissue-specific organoids representative of various tissue types for large, farmed animals. This includes mammary organoids, upper airway and small airway lung organoids, liver organoids, gastric organoids, and various intestinal organoids. These platforms are enabling our researchers to perform precision-based research that improves our understanding of host physiology, host-pathogen interactions, and drug and vaccine delivery, which will ultimately lead to new developments for the improvement of animal health and welfare. We are now using these organoids to model host pathogens' interactions in the lab. This is providing unparalleled insight for how infectious agents interact with and infect specific host tissues at disease interfaces. Next, we will demonstrate a simple method for the cultivation of gastrointestinal organoids in the lab from tissue containing primary stem cells. Cut the tissue longitudinally to expose the epithelium. Use a sterile microscope slide. Gently press to scrape off mucus. Discard as much mucus as possible. Next, press more firmly to scrape off the epithelial layer. This layer contains gastrointestinal glands or crypts, where the primary stem cells are located. Transfer the scraped epithelium to a falcon tube containing HBSS. Centrifuge the material for 2 minutes to pellet. After centrifugation, an epithelial pellet will be settled at the bottom of the tube. A grayish layer of mucus might be identifiable above the epithelial pellet, followed by the liquid supernatant above this. Remove and discard the supernatant and mucus layer. Keep the epithelial pellet. Repeat step 4 until no mucus layer is visible following centrifugation. The clean epithelial pellet can be immediately cryopreserved and stored for tissue digest at a later date. Alternatively, it can be subject to tissue digest to release glands or crypts. For tissue digestion to release glands or crypts, resuspend the epithelial pellet in digest media. Place the digest sample in a shaking incubator and gently rock the sample for 30 to 40 minutes. Place the sample horizontally, not vertically, such that the material is moving in the liquid suspension throughout the tissue digest. While the tissue digest takes place, place Matrigel on ice. We will come back to this later. Remove the tissue digest sample from the shaker and let the large material sink to the bottom of the tube. Transfer the supernatant to a new falcon tube. Make sure not to collect any of the larger debris that has settled to the bottom of the tube. The supernatant fraction contains the released crypts. Transfer an aliquot of the supernatant fraction to a petri dish and assess the sample by light microscopy for the presence of crypts. It should look like this. Centrifuge the gland or crypt containing supernatant to pellet the crypts. Remove the supernatant and resuspend the crypt pellet. Transfer an aliquot to a petri dish and count the number of glands or crypts in a certain volume. If necessary, adjust by adding more media if the glands or crypts are too dense, or centrifuge and resuspend in a smaller volume if they need to be concentrated. Transfer sample to the Matrigel that was placed on ice earlier. Mix the sample by pipetting and transfer to each well of a 24-well tissue culture plate, creating domes. Carefully transfer the plate to an incubator and leave for 20 to 30 minutes to allow Matrigel domes to set. Add organoid growth media to the wells and antibiotics. Place the sample back into the incubator. Glands or crypts should form closed organoids overnight like this. Replace media with fresh media every 2 to 3 days. After about 7 days, organoids should be well developed. Intestinal organoids typically have a branch structure. Passage organoids every 7 to 21 days. Please see associated publications for details on how to passage organoids and their utility in veterinary research.